Hi guys, so today we're talking about cellular respiration and we'll tie that in with fermentation in the next few videos. So cellular respiration is what gives organisms the amount of energy that they need to do work. Living cells require energy from outside sources. Some organisms that we know as producers are able to make their own food, while most animals, like a chimpanzee, obtain their energy by eating plants and other organisms in order to obtain that energy. Energy flows into the ecosystem first as sunlight, and then it leaves the ecosystem as heat or radiation. Photosynthesis generates oxygen and organic molecules such as glucose that are used in cellular respiration. Cells use this chemical energy stored in the organic compounds in order to generate ATP, which powers cellular work. So here we have our light energy entering into the ecosystem. It goes into photosynthetic organisms. That makes organic molecules and oxygen that are used by the mitochondria to produce ATP. It leaves as heat. And then cellular respiration makes carbon dioxide and water that are used in photosynthesis. Chapter 9.1 is all about how catabolic pathways yield the energy by oxidating organic fuels like glucose. Several processes are central to the cellular respiration and their related pathways. These are catabolic pathways, and their job is to produce ATP. The breakdown of organic molecules is an exergonic reaction. Fermentation is one of the ways that we can build ATP, and this is the particular degradation of sugars in the absence of oxygen. Aerobic respiration is what we typically think of when it comes to cellular respiration because it consumes organic molecules with oxygen in order to yield a great deal of ATP. Anaerobic respiration does the same thing as aerobic respiration, but it is in the absence of oxygen, like fermentation. Cellular respiration includes both aerobic and anaerobic components, but is often referred to as aerobic respiration because it requires energy in order to happen. Although carbohydrates, fats, and proteins are all consumed as fuels, it is helpful to trace cellular respiration using the sugar glucose. This is our equation for cellular respiration. We have 1 molecule of glucose plus 6 molecules of oxygen will yield 6 molecules of carbon dioxide, 6 molecules of water, and energy in the form of ATP and heat. This is a redox reaction, so the transfer of electrons during chemical reactions releases energy stored in the organic molecules. This release of energy is ultimately used to eventually synthesize ATP in the electron transport chain. Chemical reactions that transfer electrons between reagents are called oxidation reduction or redox reactions. In oxidation, a substance loses its electron and becomes oxidized, while in reduction, the substance is gaining an electron or is reduced, meaning that the amount of positive charge is reduced. Here's your standard redox reaction. So we have sodium and chlorine, and we get that transfer of electrons between the two. Sodium becomes oxidized because it loses an electron, and chlorine becomes reduced because it gains an electron. Just again, another way of looking at it, that movement of the electron from one to the other. The electron donor is called a reducing agent, whereas the electron acceptor is called the oxidizing agent. Some redox reactions do not fully transfer the electrons from one ion to the other, but it changes the sharing of the electron in the covalent bond. This is most commonly seen between methane and oxygen, which we see here. If we're looking at the actual electron structure, when that happens, our sharing of electrons has shifted, still doing a redox reaction. During cellular respiration, the fuel, like glucose, is actually what is being oxidized, and oxygen is being reduced into water. So this is that redox reaction using our cellular respiration reaction. Our glucose sugar becomes oxidized into 6 molecules of carbon dioxide, and our oxygen gets reduced to 6 molecules of water. A stepwise energy harvest using NAD+ in the electron transport chain. Cellular respiration uses glucose and other organic molecules to break down these molecules in a series of steps. Electrons from the organic compounds are usually transferred to molecules of NAD+, which is a coenzyme that's job is to carry electrons from one part of the reaction to another. As an electron acceptor, NAD+ functions as an oxidizing agent during cellular respiration. Each NADH, which is a reduced form of NAD+, represents the store of energy that is trapped inside of it in order to synthesize ATP. This is NAD. We don't need to know the structure. I just need you to know that NAD is for cellular respiration. When it is reduced, it is NADH. NADH passes its electrons to the electron transport chain. Unlike in a controlled reaction, the electron transport chain passes those electrons through a series of different proteins instead of causing one explosive reaction to happen. The difference is we get a slow release of energy over time versus one big burst of energy, and then our cell doesn't have any energy left to do work. Oxygen pulls the electrons down the chain in an energy yielding tumble to create water at the end. This energy yielded is used to generate ATP. So this is what we don't want, that one big explosive release of heat and light energy, but slowly over time, we spin it through a series of proteins to slowly release the control of ATP. There are 3 major steps to cellular respiration. First is glycolysis. Glycolysis breaks down glucose into 2 molecules of pyruvate. Then the citric acid cycle, which is also called the Krebs cycle, completes the breakdown of the glucose. And then we have oxidative phosphorylation, which accounts for most of the ATP synthesis. So color-coded in this tealy color, we'll see glycolysis. In red, we'll see the citric acid cycle. And in violet or purple, we'll see the oxidative phosphorylation. So out in the cytoplasm is glycolysis when glucose becomes pyruvate. We move that pyruvate into the mitochondria where it becomes acetyl-CoA, into the citric acid cycle. Our electron carriers bring the electrons down to the electron transport chain where we produce the most amount of ATP. The process that generates the most amount of ATP is that oxidative phosphorylation or electron transport chain step, because it is powered by the redox reactions. Glycolysis and the citric acid cycle will each create a few molecules of ATP, but it's not enough to really give our cells the amount of work that they need. 90% of the ATP generated in cellular respiration comes from oxidative phosphorylation. A smaller amount of ATP is formed, and that is called substrate level phosphorylation during glycolysis in the citric acid cycle. For each molecule of glucose that is broken down through cellular respiration, the cell can make approximately 32 molecules of ATP. And it happens, we have our substrate comes in that has a phosphate group attached. The enzyme just helps that ADP become ATP, and our product here is water. Glycolysis harvests chemical energy by oxidizing glucose into 2 molecules of pyruvate. Glycolysis means the splitting of sugars, and it breaks down glucose into 2 molecules of pyruvate. Glucose is too big to fit into the mitochondria the way it is, so we have to break it down into 2 3-carbon molecules that can pass easily into the mitochondria. This occurs in the cytoplasm because of the size of glucose, and it has 2 major phases. First is an energy investment stage where we need ATP in order to break glucose. And then we get an energy payoff phase where we get 4 molecules of ATP. But because we used 2, we say that we net 2 ATP in this process. Glycolysis can occur whether or not oxygen is present, meaning it is both an anaerobic and an aerobic process. Here is your energy investment stage. ADP, because we use 2 ATP, glucose gets broken down. And our energy payoff phase, we get 4 molecules of ATP and 2 molecules of NADH to result in our 2-pyruvate at the end. You don't need to know these intermediates here, but just to show our glucose comes in, we use 1 molecule of ATP right away to add in a phosphate, and we break it down. We continue to break it down. We get 2 molecules of NADH. We make 2 ATP here and 2 ATP here, but because we have already used 2, we only net 2 molecules of ATP.